All right. So your microscopy lab is due tomorrow. Okay, that was kind of the intro to using a microscope and being able to find stuff and make lab diagrams. This lab here, it's now, I got to be able to look at a cell and kind of figure out what it is I'm looking at. And I hate to say use your imagination because that sounds bad, but you might need to use your imagination a little bit when you are looking at a cell and going, I wonder what that is. I wonder what that is. Okay, don't wonder. If you think it's something, that's probably what it is. Okay, realistically, when you're looking at cells under the microscope, and some of the cells we look at today are going to be pretty small, you should still realistically be able to see a cell membrane because you can always tell where the cell begins and ends. Okay, if you're looking at a plant cell, you don't always see the cell membrane, but you see the cell wall because it's a bit thicker. Okay, you should always be able to see the nucleus. Realistically, probably the nucleolus as well, right? Most of the time, you should be able to see rough ER. It's usually around the nucleus. Okay, and what I always tell people is when you look at the nucleus, look kind of around the edges of it. It's not going to be a perfect circle, and sometimes it's going to look like it's got a beard. Okay, like on one side, if it was a face, there's always this kind of blurry, fuzzy thing that seems to be on one side of it, and that's usually the rough ER. Okay, so when you're looking at stuff, you can kind of use your reasoning, I guess, rather than use your imagination to figure out what things are. Now, if you're labeling ribosomes, I'm going to call you on that one. You can't see ribosomes, okay? You don't have an electron microscope, so you're not going to see those, okay? Things like that. But, I mean, for the most part, you should be able to see the bulk of the kind of big structures that we've talked about in class. Everybody follow me there? Okay. So the problem we're looking at today has to do with the second point of the cell theory, and that is that cells have to carry out the basic functions of an organism in order to survive. Okay? Thus, they must have structures within their cells that carry out these basic functions. Okay? So for our variables, okay, you are going to be looking at a plant cell, an animal cell, and a cell of your own. Okay? We are going to take a swab of the inside of your cheek, like they do on CSI, okay, with a little Q-tip, right? And then you're going to smear that onto a slide and make a wet mount like you did with the letter E, and you will be able to see some of what you ate this morning and some of your own cells. Yes, regardless of how well you brushed your teeth or rinsed your mouth or whatever, you are still going to have some little pieces of detritus, okay, from what you ate because it sticks to your cheeks. Human blood is also in there, but it's not part of the analysis. Just everyone always asks, can we look at blood? And so I just put it in there because everyone always asks. So yeah, you can look at human blood, but it's not going to be one that you're going to do a drawing of. Okay, so no drawing of the human blood, and no, you're not going to include it in any of your questions for the analysis or any of your answers for the analysis questions. Okay. No. Yes, you're going to do drawings of the other three. Okay. All right, so manipulated variable here, guys. We're looking at cells from different kinds of organisms, agreed? Okay, hopefully they have some things in common, I don't know. Yes, I'm sorry, you are taking pictures. That's what I mean by drawing, sorry. You're making lab diagrams with, yeah, same way you did the last time. Yeah, don't draw it, take pictures. Okay, so yes, for our manipulated variable, we are looking at cells from different types of organisms. They're all eukaryotic, that's important, okay? But they are all from different organisms. Okay, so the problem here is about carrying out basic functions and having certain structures. I would say that probably has something to do with your responding variable, okay? Because your responding variable is always what it is we're looking for. Okay, we're looking at cells from different organisms. What are we hoping to see in there? Okay, well, that was a rhetorical question. Okay. And then we need how many controlled variables? 3. I gave you one already. These cells are all from what kind of organisms? Eukaryotic. That's important because if we were looking at any prokaryotic cells, we wouldn't see any of the organelles. Okay, because prokaryotic cells don't have a nucleus or any of that kind of stuff. Okay, so important, one controlled variable, that they are all from eukaryotic organisms or they are all eukaryotic cells. It's in your notes. E-U-K-A-R-Y-O-T-E-S, eukaryotes. Okay, Hypothesis. In the if part. What should we talk about in the if part? What do we think cells do? Okay, I'd say that's pretty good. Okay, I would say the if part needs to talk about, yeah, exactly. What do cells do? Maybe the second point of the cell theory is important here. Okay, and part describes the experiment. What are we doing? Okay, same as always. All right, go back to the problem for the then part as well. Okay, they must have certain structures within their cells. Maybe like which ones? And maybe why? Okay, would be kind of an important thing to write in the then part. As you can see, I'm giving you less and less here all the time, right? Kind of weaning you off, having me do it for you. Okay, so obviously the procedure is going to be pretty much the same as last time, except different slides. Okay, so you'll get your microscope, and here's the order in which we're viewing things. Okay, you're going to be looking at the monocot dicot cross section. That's the plant cell. That one you're looking at first. Okay, it's the biggest one, and they're really good. Like you can really see a lot of stuff within the plant cells. Okay, the second one is the generalized animal cell. And the generalized animal cell is basically a sample of what's called stratified squamous epithelium. If you've ever dusted your knuckle, it's that red skin underneath that hurts a lot. Okay, that's your stratified squamous epithelium. Yeah, dusted your knuckle, like you hit it on something and it scrapes the skin off. Yeah. Okay. All right. So that'll be the second one. Yeah, and I'm going to give you an idea of kind of what they look like here in a minute. But the generalized animal cell is small. Okay, now each individual cell is small, but you are looking at a piece of skin that's fairly large. Okay, it takes up a good section of the microscope slide, so there's lots on there. You have to find a spot on there that's got a good cell that you can take a picture of. Everybody follow on that? Okay. And then the last one you're going to look at is your cheek cell. Okay, so the third one will be your cheek. Make sure, since we're going to be putting stuff from inside our mouth on the slide, that when you're done with the slide, it's washed with soap and water, please. Okay, so go back to the sink, get soap and water, clean it off, dry it off before you return it. Remember, the cover slip goes in the garbage. Okay, it doesn't get reused. Okay, and then the last one, when you have time, okay, is the human blood smear. All right, and it's pretty easy to see. Again, it's not something that we're including in the analysis. It's not something you're doing a diagram of. Okay, so this is just for fun. Okay, it's not part of the lab itself. It's just because I always get asked, can we look at blood? And rather than have someone just do it on their own and prick their finger and then someone gets hepatitis, it's better to just have you look at the prepared blood slides that we have in the lab. Incidentally, please, the only thing you can look at from your body under the microscope is your cheek cells. Okay, we don't want, well, I suppose you could pluck out a hair if you wanted to, but okay, just no like other bodily fluids, please, because that's just gross. And there's lots, well, it's a health issue. Okay, like I'm not prepared to handle blood around here. Not that I couldn't if one of you cut yourselves, but I don't want to have it out if I don't have to. Okay, all right. Okay. Now it says on here toothpick. I used to use toothpick, but for exactly the reason I just described to you, I don't use a toothpick anymore. Okay, because I had people that were like jabbing their cheeks with the toothpick, and then they're like, Mr. Clare, what's all this red stuff on the slide? Blood. Yeah. Okay. Don't. Yeah. So we just use a Q-tip now. Okay. Q-tip still gathers cells, but doesn't. Yeah. Luckily, I never had anyone have the toothpick go through, but they jabbed themselves good enough that they drew some blood. So don't do that. Okay, so for the analysis, there's only one question here, guys. Okay, there's only one analysis question, but I'm going to want a fairly thorough paragraph-style answer here. Okay, so based on your knowledge of cell structure and your observations of different cells in this lab, what organelles are most important for a cell to function, and which ones are specific to a certain mode of nutrition? Okay, so you're kind of comparing and contrasting the cells you're looking at. You've got 2 cells from an animal, 1 cell from a plant. Okay, what do they have in common, right? What are some things that are different based on what you saw? All right. I mean, yes, you could say, oh, well, in this and this and this, but if you don't see that stuff, this really shouldn't be part of your answer. Okay, based on what you saw, right? What can you say? All right, so guys, that's one question, 5 marks. Make sure your answer is worth 5 marks. If you just go, they were kind of the same. How many fingers am I holding up? Yeah, that's what you'll get. All right, so don't do that. All right, for the conclusion, remember, copy and paste your hypothesis, and then again, look at your analysis, look at your observations. What did you see? Okay, can you confirm, accept or reject your hypothesis based on the observations that you made and the analysis that you made? Make sure you talk about those things in there. And then sources of error. Okay, so when you're looking through the microscope, okay, try and think of a few things that maybe made the lab difficult, okay, that could be improved, right, for the next time. Yeah, let's hope that no one was bleeding the whole time. That's not a source. That's a human error anyway, not a procedural error. Okay, so when we're looking through the microscope, okay, generally, okay, this is some pictures I took a few years back, but I have like a 4K video again now for this one that just came up this morning, right? So when you're looking at the plants, okay, this is what you're going to see, right? We're going to be looking at, so when you open up the slide, or when you have the slide for the plant cell, okay, so there'll be the circle here in the middle. There will be 2 kind of things like that on the slide. You want to look at the bottom one, because one is from a monocot and one is from a dicot. Monocots and dicots have different leaf structures. They're different kinds of plants. Okay, we want to look at the bottom one because that's the dicot. Okay, now what you can see here are basically the different cell layers. Okay, up here up top, these are the epidermal cells. You don't see much in them. Okay, basically their job is just to secrete the waxy covering that covers the leaves. Okay, the cells you want to be labeling are the ones that are here. There's 2 layers of them in the palisade layer. Okay, so the cells are very rectangular in here. Okay, you can see them quite clearly. You can see some nuclei, like they're pink. They stain pretty well. See the little gray kind of oval shaped things? What do you suppose those are? No, they're grayish green. Okay, and they're big, and there's lots of them, so you should be able to see them pretty easily. All right, so when you're looking at that, this is the section that you want to label. Okay, pick a cell in that section to do your labeling on. Obviously, you're going to have more than one cell in your picture. If you need to label from more than one cell, then go ahead. Okay, as long as the labels are correct and all to one side and all that kind of good stuff. Everybody follow there. Okay. When you look at the animal cell, okay, the stratified squamous epithelium is it's thick. It's a thick layer of cells, and you can kind of see each one of these kind of dark dots is a nucleus. Right, so there's lots and lots of them. When you blow it up a little bit bigger, put the pencil down. Okay, this is what you'll see. All right, now you can see individual cells fairly clearly here now. Look, each one of these things here is a nucleus. Right, so it looks kind of like when mud dries and it kind of cracks. Okay, each one of the kind of cracks would be a cell. So you'd be able to see the cell membrane. Okay, and be able to see on this one here, on this nucleus right here. See how it kind of has a beard? Yeah, okay, you got to use your reasoning / imagination a little bit. Okay, these are the things you really got to look hard to find those structures. If you can, but you probably won't be able to get them all off of one cell. Yeah. Okay, like when we used to do this by hand and draw them by hand, you'd draw one cell, but you were really drawing a conglomerate of, well, I saw it on this one over here. I'm just going to put it in the one that I'm drawing because no one would know the difference, right? So now you've got many cells. Label what you can using as many cells as you need to. Okay, Kevin. That's entirely up to you. I kind of gave you an idea of which ones you should be able to see earlier there today. Okay, so yeah, it's going to be a bit tricky when you're looking at that. And remember that on the Underneath the stage, there's that condenser diaphragm that you can adjust how much light is coming through. When you get up to the high power, especially on the cheek cell, but more also on the animal cell, if you adjust that a little bit, sometimes it improves the contrast. That's the differences between light and dark, and it makes the cells stand out a little bit better. So it's something you might want to try for that one. All right, and then for the cheek cell, okay, that's my cheek cell. Pretty proud of it. It's pretty awesome. Okay, but yeah, so they aren't stained. That's the trick with the cheek cell. That's why we're looking at it last. They're transparent. If you're looking at something that's like thick and dark, you're looking at a piece of food. Okay, so if it's thick and dark, it isn't a cell. Right? Sometimes you get clumps of cells together because I mean they're coming off. Sometimes they just come off together. But you should be able to isolate and find, you know, a few cells. The key when you're making the wet mount is that when you take the Q-tip, okay, if my hand is the slide, you want to rub the Q-tip across and be twisting it while you do that. Okay, so make kind of a back and forth zigzag pattern up and down one time, okay, on the slide while twisting the Q-tip. Okay, that will make sure that you spread the cells out fairly well so that they're not in big clumps on the slide. Okay, everybody follow that idea, that procedure? Okay. Okay, so find an individual cell and be surprised. Once you get the contrast right, you can see quite a few structures within your own cell, even without it being stained. Okay. Questions on what we're doing there? All right. Try and get into fairly small groups if you can, like 3 or less, so that everyone gets a chance to use the microscopes and things like that. Okay, we have a little bit more time today than we had the last time, so we shouldn't be pressed for time. Jackson. That question's already been asked. You label as many as, like, use as many cells as you need to label the structures you think you can see. Yeah. Okay, and guys, make sure that when you take your picture, that you take it on the highest resolution possible on your phone. That way you can blow it up a little bit more when you're making the lab drawing, and you'll see a few more structures. But take it on the highest resolution that you possibly can. All right, let's head across. All right, so you've got a few things coming due here in the next little while, and we'll have one more in-class activity this week. That one will get done in class because you'll have a real full class period to do it. Okay, but you are going to have quite a bit of stuff coming up here in the next little while. It just comes kind of fast and furious here for a week or so, and then it'll cool off again for a bit. So this one's due October 30th. Okay, start of class. That's Tuesday, right? Monday. Sorry, Monday. It's Monday. Okay, so make sure you got it ready for then. Okay, and your microscopy lab obviously is due tomorrow, so make sure you have that done too. Okay, but you got some time right now. I'd be working on it.